Let's begin. This looks familiar. This is our SL equation where I told you that we're not making anything really in photosynthesis. We're just transforming these carbon dioxide and water into glucose, right, with the help of sunlight. Remember that? I pointed where did this come from? Where did this come from? We're just transforming these molecules with the help of sunlight into this. So before we understand the process, what is our essential question, anyone? Light dependent and light independent stage. All right, let's start first. Same with cellular respiration. Let's start first with getting to know the chloroplast. Because if cellular respiration happens in the mitochondria, photosynthesis happens in the chloroplast. So this is a model of your leaf sideways. This is the top, this is the bottom. If you have observed, if you take a sample leaf, any leaf, if you look at the leaf, cafeteria. If you look at a leaf, is it much greener on the upper side or the underside?
Upper.
Here in my diagram, if you look at the leaf sideways, you have a layer of cells here, again, the palisade layer. And if I zoom in one cell, let me take one cell. This is how one cell would look like. One cell has these little circles we call chloroplast. If I take one chloroplast, there I zoom it even closer. This chloroplast looks like a stack of pancakes. That's at least in my brain. That's how I imagine this because I love food. You have a stack of pancakes, and it's enclosed in two membranes. Electron micrograph, it looks something like this. And if I zoom in this pancake, this is what I see, a membrane. I see a membrane. So let's zoom out again. Leaf, palisade layer, right? You take the cell, it looks like this. You take the chloroplast, it looks like this. They have stacks of membrane here. If I take one pancake or stack of membrane there, it will look something like this one, a membrane. Now this is how your chloroplast would look like. The structure of the chloroplast is not in your book, so please listen. Very unique because it has an outer membrane, an inner membrane, and another specialized membrane here. So we're actually looking at three membranes, three bilayers. If I zoom this in, I'm going to draw something like this. So outer membrane, inner membrane, thylakoid membrane. Stack of thylakoids, we call it granum. Fluid outside the granum, stroma. Outer, inner, thylakoid membrane, granum, the whole thing, stroma, the fluid here. Also take note, if I get one pancake, because we're going to be focusing on one pancake this morning. If I get this one pancake and I zoom it in, it'll look something like this. So this is your pancake, and there's also going to be a space inside the pancake. So we call this thylakoid space. Oh, nothing fancy, it's just the space inside the thylakoid space. So thylakoid space. What's the space in the chloroplast? Stroma. Okay. Are we clear? Or if you're drawing a diagram in your notebook, that may help you understand my different stages of photosynthesis later on. Out there about the structure, let's go to the first stage of photosynthesis, the light-dependent stage. Common sense, grade 9 knowledge, dependent to light, meaning it happens in the presence of light. Okay. Allow me to simplify first. And then, music please. Dun, 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 dun. Alright. So let's do, once upon a time, there was sunlight. Okay? Sunlight hit the thylakoid membrane. It hit the thylakoid membrane. Now there are special areas in your thylakoid membrane where chlorophyll is concentrated. These areas, we call them photosystems. Photosystem 2 and photosystem 1. So again, these photosystems contain concentrated amounts of chlorophyll. Remember, chlorophyll can absorb blue and red except green. So light strikes this photosystem 2. Let's start with photosystem 2. When light strikes photosystem 2, the chlorophyll pigments there become energized. There's light, right? They become energized. They're so hyper that they lose their electron. They're energized, they lose their electron. So that electron was transferred to plastoquinone, PQ. It's in your reading. I hope you read that. So the electron was transferred to plastoquinone. This plastoquinone passes the electron to other protein carriers. You don't need to know the name. You don't need to know the name. And then eventually it gets into the photosystem 1. Again, light strikes photosystem 2. Chlorophyll pigments lose their electron. It's passed on to plastoquinone. Then eventually it goes to photosystem 1. What's so special about that? What's so special if they lose their electrons? If they lose their electrons, and if they move from carrier to carrier, we've learned this before. If a protein has an electron, it becomes energized. So as it moves from here to here to here to here, these proteins are now energized to pump protons from the stroma into the thylakoid space. This should sound familiar because this is the exact mechanism in electron transport chain. As electrons move, the proteins pump protons. Okay? So as electrons move from here to here to here to here to here, these proteins pump protons into the thylakoid space. Okay, we're clear there. Now, this chlorophyll, the chlorophylls here lost electrons, right? So it needs to be stable. They lost an electron. They're so hyper, they lost an electron. They now have a high oxidizing ability. What is oxidation again? Okay. They have a high oxidizing ability. It means they will allow water to lose electrons. So they will oxidize this water. Water will lose. Okay? So if water splits and loses electrons, the electrons goes here in the photosystem 2. So it stabilizes photosystem 2. Okay. Children, my story generated what products? Protons here, right? Also oxygen. That's how oxygen is produced. Question with that first part of the story. The next part of the story. Now, photosystem 1 is also hit by light. Actually, they're both hit by light. So this photosystem will also lose electrons. These electrons will transfer to ferredoxin. It's also in your book. And this ferredoxin will pass that electron to NADP plus reductase, another protein. And this enzyme passes that electron to an electron carrier. Our electron carrier this time is NADP plus. So this electron will go to NADP plus. In the presence of protons, NADP plus electrons plus protons becomes NADPH. So in my second story, what is produced? NADPH. Now, I want to do it like this. The chemical reactions involved here are the following: photoactivation, photolysis, photophosphorylation. Please list it down. Photoactivation, photolysis, photophosphorylation. So let's go back to my story. Where is photoactivation in my story? Photoactivation is when the chlorophylls in the photosystems loses electron. That's called photoactivation. They're activated. They're hyper. Where is photolysis? Because they're activated, what are we splitting here? Water. Why is it called photolysis? Because when light strikes photosystem 2, remember this lost electron? This has now a great oxidizing ability. The chlorophylls here have great oxidizing ability. They oxidize water, or they split water into 2H and then 2 electrons and oxygen. That's why it's called photolysis. Photo here, photo, and then this one lyses the water. So photo strikes the chlorophyll. Chlorophyll now can split water, lysis. Photolysis, photolysis. Okay? Now, where is photophosphorylation? Remember, you've learned 2 kinds of phosphorylation. I don't think you remember. What are the 2 kinds of phosphorylation you've learned? Substrate level phosphorylation and oxidative phosphorylation. Now, this one is photophosphorylation. It just means something is phosphorylated because of light. Now, how is this photophosphorylation? Because light strikes here, electrons move. These proteins have the ability to pump protons in the thylakoid space. That can establish a gradient. Looks familiar? ATP synthase.
Yeah.
So you form a gradient here. Again, they move out through the ATP synthase. ATP synthase is energized. You form ADP to ATP. Okay? Clear? Because you run out of this. This whole story tells you that the product of light-dependent stage are the following. Oxygen. Check. You know how oxygen is produced? Second product, ATP. Third product, NADPH. So oxygen will be a waste product. Light will not use it. But this too, NADPH and ATP, will proceed to the next stage of photosynthesis. Please don't forget, you need to know plastoquinone. That's the first electron acceptor. I forgot. You also need to know plastocyanin. That's the protein that will pass the electron to photosystem 1. And then, of course, pyridoxine. Plastoquinone, plastocyanin, pyridoxine. PPM. Plastoquinone, plastocyanin, pyridoxine. You will not remember, so write it down. Plastoquinone, plastocyanin, pyridoxine. So plastoquinone is the first electron acceptor. Plastocyanin is your protein that passes the electron to photosystem 1. Then pyridoxine is your first electron acceptor from photosystem 1. Okay, congratulate yourself if you understood that. Now, second stage, light-independent stage of photosynthesis. So this doesn't really need light, but it kind of still needs light because it will use the products of the light-dependent stage. So if the light-dependent stage is not making ATP and NADPH anymore, that can also limit light-independent stage. Okay? So, as we've mentioned earlier, these are the final products, right, of light-dependent stage, ATP and NADPH. And this ATP, remember, goes out into the stroma. Here. This is the stroma, right? Fluid of the chloroplast. And NADPH is also in the stroma. So guess what? Where does light-independent stage happen? Stroma. So this happens in the stroma. The light-dependent independent stage is also called the Calvin cycle. It was Melvin Calvin who not discovered, who proposed a model on how this process happens. Okay, he proposed a model, and this is to summarize the Calvin cycle. Looks familiar, like the Krebs, right? Cell respiration, Krebs cycle. All right, so the Kelvin cycle is also called carbon fixation, or not also called. The process of the Kelvin cycle is carbon fixation. The process of light dependent is photoactivation, photolysis, photophosphorylation. The chemical process or chemical reaction for light independent is called carbon fixation. It just means you have a molecule and you fix carbon into that molecule. You add carbon to that molecule. So what molecule are we talking about? So we have a free-floating RUBP molecule. That's a 5-carbon molecule, RUBP, ribulose biphosphate. You don't need to know the complete name, just RUBP is fine. Using an enzyme called rubisco, this is ribulose biphosphate carboxylase. Again, you don't need to know, you just need to know rubisco. Using this enzyme, carbon dioxide is fixed onto this 5-carbon molecule. So 5 plus 1 is 6. But look at this one. That 6 molecule is not stable, so it immediately splits into 2 3-carbon molecules. This molecule is called glycerate 3-phosphate. Okay? Now, glycerate 3-phosphate to triose phosphate, to be able to transform this to this, we need ATP. We need NADPH. Where did this come from? Light dependent. So this is why we need light dependent. Because the products of light dependent, which are ATP and NADPH, is used to convert glycerate 3-phosphate into triose phosphate. Okay? And then, one of the triose phosphate will go out to be used to make glucose. This other triose phosphate will be used to regenerate RUBP. Okay? One goes out to make glucose, the other one goes back to form RUBP again. So here is the magic, wonderful, confusing question. 3-carbon, how does that become a 5-carbon RUBP? Okay? Think with me, please. The Calvin cycle needs to happen 6 times to be able to generate 1 glucose molecule and 6 RUBPs. So RUBP, in other words, is used and consumed at the same time. Consumed and formed at the same time, the Calvin cycle. Miss, what do you mean? Okay, let's do 6 cycles. First cycle, triose phosphate. Second cycle, third cycle, fourth cycle, fifth cycle, sixth cycle. Okay, so it happened 6 times. So you have how many triose phosphates? 12 triose phosphates. From the 12 triose phosphates, we get this 1, 2, 3, 4, 5, 6. That's your glucose. Okay. And we also have to make, because it cycles 6 times, I need 6 RUBPs too, right? Correct? I need to regenerate the RUBPs. Okay, count with me, please. 1, 2, 3, 4, 5. RUBP, that's 1. 1 RUBP. 1, 2, 1, 2, 3, 4, 5. 2. 1, 2, 3, 4, 5. 3. 1, 2, 3, 4, 5. 4. 1, 2, 3, 4, 5. 5. 1, 2, 3, 4, 5. 6. Wow. Amazing. So do you understand, children? So it has to happen 6 times, and then you're sure that you have one glucose molecule, and you are sure that you have generated enough RUBP, so the cycle can just keep going on. Okay, so this cycle again needs ATP and NADPH. So if I have a diagram in the test, where do you use ATP or NADPH? You put your arrow between glycerate 3-phosphate and then triose phosphate. That's where you need this. NADP, H, and ATP. Okay. Do not forget this star main character here in the story, Rubisco. Rubisco enzyme. Without Rubisco enzyme, cycle will not occur fast. Like, it will take very slow to fix this carbon dioxide into your 5-carbon molecule called RUBP. Okay, you can handle this. I think this is easy to understand. But with your lifestyle, with your friends, with whatever activities you have, you will forget this in 4 days. So make sure make sure your notes is something you want to look at again. Okay, so why do we believe Calvin? What was his evidence for proposing this Calvin cycle? First of all, Melvin Calvin was able to do an experiment because 2 things were discovered during his time. First, carbon-14 isotope. Remember in chemistry, we've learned that carbon-14 isotope is radioactive. Unlike carbon-12, which is kind of stable, carbon-14 is radioactive. It degrades. It emits radiation. Okay? So because we have that discovery, also we have autoradiography. So autoradiography is just like a special type of X-ray diffraction where you can take a picture of something that is radioactive. That's autoradiography. Together with these 2 technologies together, he was able to propose an experiment on light-independent stage. Allow me to use BioNinja. Because they have a very easy to follow diagram of the lollipop experiment. It's called the lollipop experiment because the glass looks like a lollipop. Okay, so in the lollipop experiment, Calvin put algae here. Algae is a photosynthesizing cyanobacteria. So it's in the water and it's green and it can photosynthesize. Okay? And then he injected carbon-14 into the sample. So in other words, the plants will use carbon dioxide, right, in carbon fixation. This will use carbon dioxide in carbon fixation. The carbon dioxide, the carbon in the carbon dioxide is carbon-14. And you already know that carbon-14 is radioactive. So he injected CO2 using carbon-14. So whatever CO2 that this plant, this algae will absorb, will have CO2 that has the carbon-14. Okay? And then what he did was, so this was exposed to light. So hypothesis is this is photosynthesizing. So this was exposed to light. And in different time intervals, he was taking samples of the algae here. Okay? Then what did he find out? Actually, this is a video. Let's watch it. So radioactive carbon is added to algae to be used in photosynthesis. After a set period of time, the algae is killed, and then a sample was taken. Okay? This sample was separated using 2D chromatography. So it separates all the carbon molecules present. And then another chromatography, it further separates the different carbon molecules present. And then using autoradiography, I said it's similar to an X-ray. So they looked at the type of carbon molecules found in the samples at different time intervals. So at 5 seconds, they have RUBP carbon molecule. At 15 seconds, they have GP molecule. At something seconds, they have TP molecule. Therefore, he concluded that RUBP is transformed into G3P, which is transformed into TP. You may not have understood it. Let me go back to... Okay, listen. So they took samples again in different time intervals, right? In 5 seconds, a carbon molecule that was present is RUBP. After 15 seconds, they got a sample from the lollipop again. Then using chromatography and X-ray, the next molecule present was GP. After 30 seconds, the next molecule was triose phosphate. So if you were Kelvin, it just makes sense that RUBP is transformed to GP, which is transformed into TP, triose phosphate. Okay? In your book, if you look at the diagram on page 397, you have a graph there. You have a graph in 397. How did Kelvin propose that G3P is transformed into TP? Look at the graph. Something is decreasing while the other is increasing. What is decreasing through time? G3P. What is increasing through time? Triose phosphate. So if you're Melvin Kelvin, easy conclusion, right? One is decreasing while another one is increasing. So this probably is becoming this. That's how we propose G3P forming TP. Okay, do we understand that? So this was his experiment. That's the algae, they have a source of light, and then you have here where you get samples every 5 seconds, 10 seconds, 30 seconds, and then you're looking at the carbon molecule that is present. This is not possible if we don't know carbon-14 isotope. If this hasn't been discovered, how can he monitor the carbon being formed? He was able to monitor that because we can use carbon-14 isotope. This is radioactive. We can follow the emission of radiation by the different molecules. If there's higher radiation, it's probably RUBP because that's 5 carbon molecules. There's more carbon-14 there that to emit radiation. Also, autoradiography. Autoradiography is taking pictures of those spots in the paper, in the chromatograph. Yes.
How do they know if it's G3P or triose phosphate?
Autoradiography. This is a simplified diagram. So these sample pigments, if you took X-ray films of that, the molecules, well, 5 molecules is bigger than 3 molecules, right? So it will have a different diffraction pattern. So that's how they determine now this is a 5 carbon molecule, this is a 3 carbon molecule. So again, without this autoradiography, he can't really determine it. And without carbon-14, that can emit radiation, how can you diffract them into different positions? What's important for you to know here is nature of science. What's the nature of science here? You don't need to know the experiment in detail, okay? What you need to know is Kelvin was not able to propose the model for the Kelvin cycle if it
Was not for
because it's carbon-14, isotope discovery, and autoradiography. Without those two, his lollipop setup won't work at all. He wouldn't know what to study. Okay? So do we have any questions? Okay, this is much shorter than cellular respiration. Remember, at the end of the day, I told you, I've been telling you, I don't know if somebody did this already. Print this, post it on your bedside wall. Photoactivation, photolysis, photophosphorylation. Put this on your bedside wall. Why you don't do it? If you do it, can you take pictures and then like show it to me? I'll be very happy.
Yes.
There's no other way to memorize this.
You put it in the ceiling. You can just lay
All right,
lay
we'll have a
away
little
from
bit
it.
of brain break, and you need to do the second worksheet that the SLs are also working on. The pigments, worksheet for pigments.
Of course, they're Calvin cycling.
Yeah, why not?
This looks